This is part 2 of lecture 2 of Bio 310. Um, so the whole logging process summarized. You have the primase that's making the RNA primer, the DNA polymerase is making the Okazaki fragment. The RNA primer is removed, ligase is used to glue the rest of the gaps between. So no, I'm sorry. Our DNA polymerase replaces the RNA with DNA ligase seals the fragments, continuous data strand. Right, okay. If we specifically look at these little diagrams, you have the primase joining the two ribonucleotides. Primase synthesizes 5' to 3'. We're just going to be looking at the diagrams now. So you have the incoming ribonucleoside triphosphates, the template DNA strands, the primase joins together and together two ribonucleotides. Um, the primase synthesizes 5' to 3' direction. Previous Okazaki fragment, previous RNA primer, new RNA primer. Synthesized by primase. RNA polymerase adds nucleotides to the 3' end of the RNA primer to synthesize Okazaki fragments. DNA polymerase finishes the Okazaki fragment. Previous RNA primer is removed by nucleases and replaced by DNA by repair polymerase. Next sealed by DNA ligase. Okay, real quick final stretch. Um, the diagrams we're going to have to go over later. DNA proofreading and polymerase. DNA proofreading, DNA polymerase sometimes adds the wrong nucleotide, right? Example, instead of AT, it can accidentally add AC. How do we fix this? DNA polymerase notices the mistake. It stops, removes the incorrect nucleotide, inserts the correct one, continues replication. This proofreading makes DNA replication very accurate. So if there's anything wrong, it will just stop. I'll take it out and insert the correct one and start again. So why can proofreading only happen in the 5' to 3' synthesis? We know synthesizing always happens in the 5' to 3', and why can proofreading happen at that time? Because the incoming nucleotide carries the three phosphates, triphosphate. Those phosphates provide the energy to make the phosphodiester bond. So if the DNA polymerase, we've talked about this before, right? You need to break the bond between two phosphate bonds. So you can get energy from that ATP, and that is used to make the phosphodiester bond so that the nucleotide can be added. That's what we got before, right, with the helicases and everything. Um, and that was after the topoisomerase, I believe, right? Like here, yeah. No, I mixed it with the. No, it is definitely topo. Is it though? No, I don't think it's Yeah, this concept was mostly about yeah, it was the topoisomerase case. Topoisomerase release the stress, and after that, the ATP says hydrolysis, the upcoming nucleotide. Okay, back to this. If DNA polymerase removes the wrong nucleotide, then in next incoming DNTP has its own energy, so synthesis can continue. If DNA were made 3' to 5', removing a wrong nucleotide would remove the energy needed to keep extending the strand, so replication would stop. So an easy way to think about is that synthesis always occurs, DNA synthesis always occurs at 5' to 3', because proofreading can occur while still allowing the DNA synthesis. So it always goes from 5' to 3' because proofreading can also happen at the same time, and that's needed. Otherwise, you cannot really go back on that. If there's a mistake, it would remove the energy needed to keep extending the strand, so the replication would stop if we were going from 3' to 5'. So they have some proteins you have to memorize. So and what their jobs are. So helicase, we know it unwinds the DNA. Topoisomerase, we know it fixes whatever the sorry, excuse my language. Whatever the helicase does, right? To relieve supercoiling, torsional stress. Single-stranded binding proteins (SSBs) keeps the DNA strands separated. Um, That's pretty contradictory to what it's supposed to do, right? Binding, but it keeps the DNA strand separated. Primase makes the RNA primers. DNA polymerase adds DNA nucleotides 5' to 3', and then proofreads. Sliding camp holds the back to this. Yeah, this is what we're talking about. Sliding clamp holds the DNA polymerase onto the DNA so it doesn't fall off. Clamp loader uses ATP to place the sliding clamp on the DNA. DNA ligase seals the Okazaki fragments together. So helicase opens the DNA topoisomerase, removes the twisting ahead of the fork. SSB keeps the strands apart. Primase lays down the RNA primers. DNA polymerase extends DNA 5' to 3'. Give me 1 second. Right here. Um, This is what I'm talking about. So primase lays down RNA primers, DNA polymerase extends 5' to 3'. A leading strand is made continuously. Lagging strand is made as Okazaki fragments. RNA primers are removed and replaced with DNA. DNA ligase seals the fragments into one continuous strand. DNA polymerase proofreads and corrects any mistakes. So this is important. Going to be going over the bacterial replication fork. This picture shows everything happening at one replication fork in bacteria. So you have newly synthesized strand, leading strand template, DNA polymerase on leading strand, um, DNA polymerase on lagging strand, just finishing an Okazaki fragment, sliding clamp, the whole jazz. The DNA polymerase on lagging strand in process of synthesizing Okazaki fragments, lagging strand template, yada yada yada. Okay, these are the stuff we went over before. So this is like think of it as a tiny factory. The parent DNA helix, this is the original DNA that's still double-stranded before replication. So if we can find the parent DNA helix somewhere, right here, it should be here as well. Yeah, parent DNA helix, it's double-stranded, it's the original DNA. Then the DNA helicase comes along, which is yellow. DNA helicase, where are you? Right here, right here. DNA helicase comes along. It's yellow, opens the DNA, breaks the hydrogen bond, um, keeping the two strands separate. Without helicase, DNA cannot be copied, right? Then DNA primase comes along. What does it do? Turn it into whatever. It makes RNA primer. I'll forget that. Okay. Single strand binding proteins, brown. Single strand binding proteins. Where is it? These things, I think. Once helicase separates the DNA, the strands would naturally zip back together. These proteins bind single DNA strands, keep them separated, prevents knots and tangles. Think helicase zipper opener, SSB holds the zipper open. Now, where is my SSB? SSB should be around here. Where? Sliding gap right here. Clamp loader, clamp, sliding clamp. So it's holding it open. So you have the DNA, what is it? Helicase, which is opens it, and then sliding gap SSB is what holds it, keeps it open. And we know SSB's job, yeah, it's to keep the strands apart. After that, the primase comes along. Where's my primase? Primase makes the RNA primer right here. And then remember, DNA polymerase cannot start DNA, it's by itself. Primase gives the polymerase somewhere to begin. So it can begin soon. RNA primer is a very short RNA. DNA polymerase attaches here, right? Single DNA binding protein, DNA polymerase is attaching here. Um, Later, this RNA gets removed. DNA polymerase is the leading strand now. Leading strand, since DNA polymerase always builds 5' to 3', this strand happens to be oriented correctly. So, because it look 5'. Um, and then, so polymerase never stops, continuous synthesis. DNA polymerase lagging strand. Now we're to the lagging strand. This is like kind of all over the place. This is something you definitely have to go over with the professor. This is slide 8. Well, 9. Okay. 9. The template runs the RNA polymerase and the lagging strand is running in the weird way, right? It still only works 5' to 3', but the template is running the wrong way. We see that 5' is here, 3' is here. So the polymerase copies little sections, lagging strand made in pieces. Made in pieces, each piece starts with a new RNA primer. Okazaki fragments are the little DNA pieces. Eventually all pieces are connected. Sliding clamp comes again. It acts like a seat belt. So sliding clamp is not the same as SSB. I was mistaken. Sliding clamp is acting like a seat belt. It holds the polymerase tightly onto the DNA, more stability, more space. Speed clamp loader. You have the clamp loader right here. Sliding clamp cannot push itself onto the DNA. Clamp loader uses ATP, opens the clamp, places it around the DNA. Why is lagging strand looped? Notice the big loop. Where's the big loop? I guess it's this one, right? This lets both DNA polymerases move in the same physical direction. Instead of one going backwards, the lagging strand bends around. This is called the trombone model. It lets two polymerases stay attached together. So the entire bacterial replication sequence goes like this. Helicase opens the DNA. SSB protein stabilizes the strands by keeping it open. Primase makes the primer. Sliding clamp attaches. DNA polymerase copies DNA. Leading strand, continuous lagging strand. Okazaki fragments. Ligase joins the fragments. And then in the next slide, it's almost identical. The difference is that there's different DNA polymerase. If you can see, it's E and A. There are two different DNA polymerases. They work together with the primase. Its job is to make the RNA primer, add a few DNA nucleotides. After that, another polymerase takes over. Think as the alpha starting the replication. So I would say alpha DNA polymerase is starting the replication. Alpha polymerase E epsilon copies the leading strand, continuous synthesis. DNA polymerase delta copies the lagging strand, making the Okazaki fragments. CMG helicase has the same job as a normal helicase. It unwinds the DNA. Sliding clamp keeps the polymerase attached. In eukaryotes, it's called the pCNA. RNA primer is still needed, made by primase, removed later. Main difference from bacteria is that instead of, okay, we're looking at, this is bacterial, right? Prokaryotic, and this is eukaryotic. Instead of, for eukaryotic, we're seeing more diversity in like polymerases, right? There's two different polymerases. Another thing is that you have